Introducing to you Dentin and Enamel Formation, Group B1.1. Have you ever wondered how our teeth are so hard and strong? Well, it's all due to the detailed developmental process it undergoes before erupting, starting from just placodes to eventually reaching the late bell stage where histodifferentiation occurs. Two very important processes happen here, and they're called Dentinogenesis and Amelogenesis, and that's exactly what we'll be learning about today. So, here goes part 1, Dentinogenesis. Differentiation begins when inner enamel epithelial cells release tissue growth factors, insulin-like growth factors, and bone morphogenic protein, all which contribute in stimulating pre-odontoblasts to differentiate into odontoblasts, and this changes the cell's morphology from ovoid to columnar. Intracellularly, the cell becomes polarized as organelles such as Golgi complex and rough endoplasmic reticulum increase in number, and apical processes occur. The cell then deposits predentin, which is a soft organic matrix rich in glycine, alanine, and proline. This layer will be the first layer of mantle. Fibroblasts come into play and secrete the framework of thick collagen fibers. Then, fibroblasts and odontoblasts move toward the pulp, while the odontoblastic processes remain embedded in the matrix and a narrow layer of predentin is always present on the surface of the pulp. These processes secrete calcium hydroxide, which calcifies the matrix. Now, an important characteristic of dentinogenesis is its mineralization process. Two patterns of dentine mineralization can be observed, globular and linear, which depend on the rate of dentine formation. Globular calcification involves the deposition of crystals in several discrete areas of the matrix with the aid of alkaline phosphatase, which causes the pH in dentine to be alkaline. Globular masses will continue to enlarge and finally fuse, forming a single calcified mass. Small spaces in between, called interglobular dentine, occur, and they are not mineralized due to failure of fusion of mineralized globules. The remaining bulk of mineralized dentine is known as circumpulpal dentine and can be mineralized in either globular or linear pattern. In linear pattern, mineralization appears to be slower and more uniform, as shown in the picture now. Notice the difference here, compared to globular dentine, which appears less organized and does not follow any aligned pattern. Now that we covered dentinogenesis, here goes part 2, amelogenesis. Amelogenesis is the formation of enamel by ameloblasts of epithelial origin facing the odontoblast layer. Enamel formation begins at the early crown stage and involves the differentiation of the cells of inner enamel epithelium first at the cusp tips and then sweeping down the slopes until all the cells have differentiated into ameloblasts. The life cycle of ameloblasts consists of 6 stages: morphogenic, organizing, formative, maturative, protective, and finally, desmolytic. In the morphogenic stage, the shape of the crown is determined. We have the inner enamel epithelium, which is ectodermal in origin, cuboidal in shape, with a largely centrally located nucleus, and contains less organelles and cytoplasm. The Golgi apparatus is located adjacent to the stratum intermedium, while the mitochondria and other organelles are scattered in the cytoplasm, creating a non-polarized, undifferentiated cell. This cell will become a pre-secretory ameloblast during dentine formation. Here comes the cool interaction between odontoblasts and ameloblasts. The inner enamel epithelium will induce the dental papilla cells to differentiate and secrete dentine. In return, dentine will induce the inner enamel epithelium to differentiate into pre-secretory ameloblasts. This is referred to as reciprocal induction between the ectomesenchymal cells and epithelial cells. The pre-secretory ameloblasts are elongated, and the nucleus will shift towards the stratum intermedium. The Golgi complex will move towards the center, and the rough endoplasmic reticulum will increase in number, resulting in a highly polarized cell that is ready to secrete enamel. This marks it as the second stage in the life cycle of ameloblast, organizing stage. The formative stage is also known as the secretory stage. It consists of 3 parts, and the end result is the production of an enamel matrix. First part, is initial secretory stage, where the cells are columnar in shape and contain numerous secretory granules. It forms close to the dentino-enamel junction, and it contains no rods or interrods. Its granules will then migrate to the distal end of the cell and release the protein, which will attach to minerals perpendicular to the dentino-enamel junction to form initial enamel. As the first layer of enamel is deposited, the ameloblasts will move away from the dentino-enamel junction, leaving behind a pyramidal cytoplasmic extension, forming Thom's process, where it secretes rodded enamel, including inner and outer enamel, which is partially mineralized by alkaline phosphatase, and this is secretory stage. When Thom's processes form the full thickness of enamel, which is partially mineralized, it will disappear and the ameloblast will return to its original shape, short and columnar. This short and columnar ameloblast will form the enamel layer on the outermost surface of the tooth, which is also devoid of rods, marking it as the end of formative stage. Maturation stage represents the hardening of the partially mineralized enamel, starting from the growth centers, which are the dentino-enamel junction and the cusp tips. Once the immature enamel deposition is complete, 25% of secretory ameloblasts will undergo apoptosis, and the remaining will reduce in size, presenting mature ameloblasts. This is where they will modulate between two forms: ruffle-ended ameloblasts, which occupy 80% of the cycle, and smooth-ended ameloblasts, which occupy the remaining 20%. The distal end of the ruffle border is broad with tight desmosomal junctions, while the proximal end has leaky desmosomal junctions. It will then degrade the organic matrix to fragments by the acidic zone produced by the proton pump, along with the acid produced from the enamel crystal formation. The mature ameloblasts secrete lysosomal enzymes that will be targeted by mannose-6-phosphate receptors located on the ruffle border. When the acidity reaches a dangerous level, then it will modulate to smooth-ended ameloblasts, where the proximal end becomes broad with tight junctions and the distal end is narrow with leaky junctions, allowing the removal of degraded organic matrix. Once the mineralization of enamel is complete, the ameloblast further reduces in size and becomes part of the reduced enamel epithelium, which covers the newly formed crown surface. With this, we have covered the entire process of dentinogenesis and amelogenesis combined, so let's watch it in action. Notice how both processes happen simultaneously. Because these two processes are extremely detailed, common defects may occur. Common defects of dentinogenesis include fluorosis, which is fluoride incorporated into dentin during active dentinogenesis, which increases the hardness of dentin. Dentinogenesis imperfecta, which happens due to genetic deficiency of dentin sialophosphoprotein, thus showing irregular and disoriented tubules, and obliteration of the pulp chamber with abnormal dentin, resulting in a weaker, discolored, and translucent tooth, more prone to break and loss. Tetracycline has a strong affinity to becoming incorporated into bone and dentin. Exposure to tetracycline in forming teeth gives the dentin a yellow color, which after prolonged exposure to light turns grayish. Similarly, common defects of amelogenesis include fluorosis, which is caused by overexposure to fluoride during the first 8 years of life, when most permanent teeth are still being formed. Affected teeth will appear mildly discolored but highly resistant to caries. Enamel pearls. They are hard lumps of enamel. They develop on the tooth root, and they are caused by the residues of Hertwig root sheath. Amelogenesis imperfecta happens due to a genetic deficiency in the AMELX, ENAM, and MMP20 genes, which provide instructions for making proteins essential for normal tooth development, such as ameloblastin, enamelin, tufteline, and amelogenin. As a result of this mutation, the teeth are abnormally small, thin, and soft, and may have a yellow or brown discoloration. To conclude, it's important to remember that dentinogenesis and amelogenesis are very detailed processes and may be easily affected by several factors, especially during the early years of life.